In this video, I want to explain how an amino acid-based hormone is made. Amino acid-based hormones. How they are made. Okay, you've probably learned this many times in biology. And for all the fine details, you can reference a biology chapter in your textbook. There, they'll explain how the cell synthesizes proteins. I'm going to keep it very simple here. Here I have a cell. And within that cell, I have the command center, the nucleus. And within that nucleus, we have DNA. And on that DNA, we have a gene. This gene is going to code for a particular protein. So if I think of some hypothalamic hormones, we have, let's see, GnRH, CRH. We have TRH, thyrotropin-releasing hormone. Now, thyrotropin-releasing hormone is an amino acid-based hormone. How do I know that? Well, it's not a thyroid hormone, it's not T3, T4, and it's not the catecholamines, epinephrine or norepinephrine, so it's not a tyrosine derivative. It's also not made in the gonads, testes or ovary, placenta, or adrenal cortex, where steroid-based hormones are made. Therefore, it falls under the category of everything else, the amino acid-based hormones. We take that gene and we're going to first transcribe it. And during transcription, we take a gene or DNA and we convert it into messenger RNA. This messenger RNA, after we copy this, can leave through a nuclear pore. But before it leaves through that nuclear pore, we need to protect it because the enzymes in the cytosol will begin to degrade that messenger RNA unless we add a 5' cap and a poly A tail. So I'm going to put a top hat on this, and I'm going to add the poly A tail, which is going to protect this messenger RNA from enzymatic degradation in the cytosol. We don't want to be making this amino acid-based hormone all day long. We want to make whatever we need and then stop the translation of that messenger RNA into its protein. Now, where does that occur? That occurs wherever we have ribosomes. There's the large subunit and the small subunit. Now, ribosomes take messenger RNA and transfer RNA carrying single amino acids are going to assemble a sequence that forms our protein. So as this messenger RNA comes through, we get our protein. But we're not complete yet. Okay? Now, this ribosome, because this protein is destined for export from the cell, we find that ribosome on the rough ER. The rough ER is studded with ribosomes. And the folds in the rough ER are going to allow for this protein to fold properly within this protected environment of the rough ER. And when it folds properly in this environment so that it can withstand the environment outside the cell, it gets packaged into a transport vesicle. Now, this transport vesicle is going to move through the Golgi apparatus, the shipping and receiving center, which also is made up of these folds or cisterns. And as it moves through, that vesicle is going to, and the protein that is contained within it is going to be modified a little bit. We might snip an end of that protein. We might add a carbohydrate tag to it. We're going to change what's called a pre-pro hormone through post-translational modification into the final hormone. So, after translation, we have post-translational modification, where we take this protein, which in this case would be known as the pre or maybe pro hormone, and we are going to take that pre-pro hormone and change it to its final hormone. In this case, TRH, thyrotropin releasing hormone. Now, when this vesicle binds with the plasma membrane, the rough ER phospholipids that were incorporated become part of the membrane. A little bit of modified protein from the Golgi become part of the membrane, but the pre-pro hormone that was modified into the hormone TRH gets released. And for amino acid-based hormones, we know that they're water soluble. So they don't need a protein carrier in order to get right into the bloodstream build some epithelial cells here. It doesn't need a protein carrier to get into that bloodstream. It's water soluble, and blood is mostly water, so it can move right in and be carried. TRH is made in the hypothalamus and affects TSH producing cells of the anterior pituitary. It's going to get carried right to that anterior pituitary to have its effect. So, one more time, pre-pro hormone and the finalized hormone. I want to highlight one key feature here that's characteristic of these amino acid-based hormones. We have a gene for TRH. We transcribe that gene into messenger RNA. To that messenger RNA, we add a 5' cap and a poly A tail and then send it through a nuclear pore, where it will encounter a rough ER ribosome and be translated from the messenger RNA into the pre-pro hormone. Now, this pre-pro hormone gets packaged within that vesicle and undergoes post-translational modification. We snip an end here, we add a carbohydrate tag there, and we modify that pre-pro hormone into the finalized thyrotropin releasing hormone, or some people will say thyroid releasing hormone. The vesicle fuses with the plasma membrane and it gets released into the blood. But here's an interesting thing. Because they're in membrane-bound vesicles and they can't freely pass through that vesicle, TRH can be stored within the cell until the cell's stimulated to release that hormone. So amino acid-based hormones may be stored within the cells that made them. This means that the blood value, or the amount that's found in the blood of an amino acid-based hormone, does not reflect or indicate the rate of synthesis, how quickly it's being made. We're going to see that the opposite is true for steroid-based hormones. Again, you can reference your textbook for additional details if you'd like, but I'm just keeping it quite simple here.